Hello, dear friends. Welcome back to Longevity Now FL. I'm Luigi Fontana, a professor of medicine and the scientific director of the Charles Perkins Center RPA Clinic of the University of Sydney in Australia. Today, I'm excited to share with you the findings of a recent paper I published in Aging Cell, one of the leading scientific journals for the study of aging biology. This particular study looks at how the two powerful lifestyle interventions, calorie restriction without malnutrition, with optimal nutrition, meaning with all the vitamins, minerals, and fibers necessary to promote health, and endurance exercise training affect biological aging over the long term, meaning these people have been practicing this lifestyle intervention for many years. Okay, in the first part, I will break down these findings in simple terms for lay people, and then in the second part of the video, I'm gonna, instead of dive into more specific technical details for scholars or scientists who are interested in the more sophisticated data. Okay, so the key findings is that individuals practicing long-term calorie restriction without malnutrition, so these people, they are not eating just half a hamburger, half pack of French fries and half soda, because this is calorie restriction with malnutrition, and runners and Master athletes age more slowly. Yes, they age more slowly than sedentary people who consume typical Western diets filled with ultra-processed and animal foods. These data suggest that the way we live, particularly our eating and exercise habits, can have a profound impact on how our bodies age. One of the key insights from this study is the powerful effect of calorie restriction on aging markers, particularly in the blood and gut microbiome. CR, which involves reducing calorie intake with optimal nutrition, can dramatically slow down the biological markers associated with aging. However, exercise in this study shows also a major role, a crucial role, especially in slowing down the biological aging of tissues by reducing inflammation and promoting tissue repair, making it a potent complementary tool against aging. When we break down the benefits of these two interventions, we find both shared and unique benefits. The shared benefits are both CR and exercise help reduce oxidative stress and DNA damage. This means they both assist in keeping our cells younger and healthier and more capable of repairing themselves over time. These are crucial factors for aging, as cellular damage is one of the primary contributors to the aging process. The unique benefits of calorie restriction are, well, CR doesn't just help with weight loss or weight control, it also boosts metabolisms in ways that promote gut health and overall metabolic and hormonal well-being. Moreover, it provides cellular protection against oxidative damage, acting as a shield for our cells from harmful environmental factors. The unique benefits of exercise are that it offers unique benefits by calming an overactive immune system. Yes, by inhibiting an overacting immune system, which is often linked to various age-related diseases. This is very important because chronic inflammation, as you know, inflammaging, as my dear friend Claudio Franceschi has shown, is a key driver of aging and age-related illnesses. So, in simple terms, what does these data mean for you? How can you apply this knowledge to your own life to slow down your aging, your biological age, and stay healthier for longer? Okay, number one, start small with calorie restriction. You don't have to make drastic changes right away. Start simply by cutting out empty calories. All these ultra-processed refined foods and sugary drinks that are so pervasive in our modern society, and replace them with nutrient-dense foods such as vegetables, a wide variety of vegetables, whole grains, minimally processed whole grains, legumes, seeds, nuts, fish, and seafood. Over time, these can lead to significant improvements in your health and how your body ages, as I have explained in many of my scientific articles published in high impact factor journals and in my books like The Path to Longevity and the Manual to Health and Well-being. Number two, make exercise a priority. Exercise doesn't have to be intense to be effective, but it is a daily medicine. Yes, it's a daily medicine. It's not something that is working if you only do exercise, if you exercise a couple of times per week. Aim for at least 45 minutes of moderate aerobic activity per day with 20 to 30 minutes of resistance weight-bearing exercise 2 to 3 times per week. Activity like walking, jogging, cycling, swimming, play soccer or volleyball or dancing with your friends can make a big difference in your physical and mental well-being. Regular exercise not only helps you to maintain a healthy weight, but also supports your body's ability to repair itself and fight off age-related diseases. Number three, combine both interventions for maximum benefits. For the best results, combining both CR and exercise is key, is crucial. These habits work together, they reinforce each other in complementary ways to help you stay healthier and feel younger longer. When you fuel your body with the right nutrients and keep it physically active, you are giving yourself the best chance to age slowly and gracefully, as I've recently explained in an opinion paper I wrote for the European Heart Journal. That is one of the most important cardiological journal in the world, where I summarize the 10 points for healthy longevity, and there is no doubt that diet with moderate calorie restriction, with optimal nutrition and exercise are two of the most important tools to slow down aging. The bottom line is that while aging is inevitable, we do have some control over how we age. Simple changes in diet and exercise can have profound effects on your biological age, reducing it and improving your health and quality of life. By adopting healthier habits, even gradually, step by step, you can slow down the aging process and enjoy better health for many years to come. Okay, so this is the simple, non-technical summary of our study. However, for those who are more scientifically inclined and eager to explore the complexity and technicality of our findings, now let's break it down in a clear and more detailed way. As you know, recent advancement in omics technology, coupled with the development of biological age, BioAge algorithms based on molecular biomarkers, provide an exciting and unique opportunity to assess the potential benefits of different lifestyle or pharmacological intervention without the need for prolonged follow-up periods typically required in randomized clinical trial and epidemiological, randomized clinical trial in animals, because it's in humans it's impossible to do survival studies, or epidemiological studies. Accumulating evidence from prospective studies suggests that these biomarkers can effectively measure biological age and predict longevity and the risk of age-related diseases. Of course, we are still at the infancy of this science, but the data are accumulating fast and they are very promising. Cross-sectional observational studies examining molecular biomarkers of biological aging offer valuable insights into the potential benefits of long-term means more than 3 years of practicing an intervention in this study, calorie restriction and endurance exercise, overcoming the nearly insurmountable challenges of multi-year clinical trials that are not feasible both in terms of adherence and cost. So this is the best tool we have to study what happens if you exercise or you do calorie restriction with optimal nutrition for many years. It's going to be impossible to do a 7 years randomized clinical trial on calorie restriction on exercise. Impossible. Okay, so this is the best data with all the limitation of a cross-sectional study that we can have in studying the effects of this long-term intervention on aging biomarkers. As you know, many BioAge estimation methods have emerged, including DNA methylation, epigenetic clocks, metabolomics-based aging clocks, and phenotypic age, phenoage assessments using blood biomarkers. Moreover, new statistical approaches, such as the Cohen's method, allow BioAge to be computed as a multivariate distance from a reference young population, addressing batch effects and capturing nonlinear biomarker relationship. This study published in Aging Cells, explores BioAge differences among middle-aged and older adults, average age 55, who have been practicing long-term calorie restriction without malnutrition or endurance exercise, mainly runners, compared to age and sex matched healthy individuals consuming a Western diet. BioAge was calculated using multiple omic biomarkers, including proteins and immune biomarkers from Levine's PhenoAge, gut microbiome data, blood metabolomics and hormone profiles, and DNA methylation and transcriptomics from colon mucosa biopsies. A group of 6 young, aged 21 to 27 men and women, sedentary men and women, consuming Western diets, served as the reference population. The study compared PhenoAge, a composite score derived from 9 simple blood biomarkers in 41 lean men and women practicing CR for an average of 7 years, range 3 to 15 years, along with 41 age and sex match endurance runners on a high calorie diet, and 35 age and sex matched sedentary controls following a Western diet. The energy intake of the CR group was about 1800 calories. The exercise group was 2800 calories, and for the control sedentary group was 2400 calories. As I said, additionally, a subset of participants, 12 CR, 11 exercisers, and 12 sedentary controls consuming Western diets, underwent very expensive multi-omic profiling, including DNA methylation, Illumina, using Illumina 450K bed chips array, and RNA sequencing of colon mucosa tissue, blood metabolomics using targeted LC-MS, and stool metagenomics using 16S RNA sequencing. Very, very, very expensive study to perform all these measurements. For each omic layer, we calculated corresponding BioAge measures, resulting in 6 distinct biomarkers: PhenoBioAge, Microbiome BioAge, Metabolome BioAge, Hormone BioAge, DNA methylation BioAge, and Transcriptome BioAge. These BioAge measures are defined using Mahalanobis distance relative to the average profile of the Young reference group, as implemented in the BioAge R package. By utilizing a Young reference population, we make sure that precise comparison across diverse omic profiles were possible, addressing the lack of standardized definition from some BioAge measures, for example, gut microbiome, and mitigated bias induced by batch effects. So here what we found, very, very interesting data. Well, a comparative analysis of the BioAge measures across the CR, exercise, and WD groups revealed significant differences. So this is the first finding. There were significant differences between CR, exercise, and WD. Notably, individuals in the CR group exhibited significantly lower values than those in the sedentary control group for PhenoBioAge, Microbiome BioAge, DNA methylation BioAge, and Hormone BioAge. Similarly, the exercise group showed significantly lower values than the sedentary control group for DNA methylation BioAge, transcriptome BioAge, and hormonal BioAge. Interestingly, there was a significant difference between CR and exercise, with CR participants displaying lower PhenoBioAge in comparison to exercise. However, endurance exercise, interestingly, exhibited lower DNA methylation by age values relative to the CR practitioners. Very, very interesting data. For sensitive analysis, we investigated 3 additional biomarkers: the original Levine's PhenoAge, the Horvath epigenetic clock, and the epigenetic mutation load EML, using DNA methylation data from colon mucosa. Our analysis revealed significant differences in PhenoAA, defined as the residuals of the linear regression of PhenoAge on chronological age. Again, PhenoEA is defined as the residuals of the linear regression of PhenoAge on chronological age. Between the CR and WB group, between the CR and the sedentary control group, there was a difference of 2.96 years, highly significant. So basically the CR individuals were 2.3 years younger than the sedentary control group consuming Western diet. with no significant difference comparing exercise with the sedentary control group. Similarly, the Horvath DNA methylation AA was significantly lower in the exercise group by 2.25 years compared to the WD, but not significantly different in the CR versus the WD comparison. Furthermore, the EML age acceleration measure showed significantly lower values in the exercise group relative to both the WD minus 4.9 years and the CR group minus 4 years. Very, very interesting and, to be honest, unexpected data. The results of the enrichment analysis of the candidate inflammatory pathways based on DNA methylation and mRNA omic layers revealed that the calcium signaling and reactive oxygen species glutathione cytotoxic granules pathways were significantly enriched in the CR signature, highly significant. In contrast, the apoptosis signaling, phagocytosis, antigen presentation, and complement cascade pathways were significantly enriched in the exercise signature. These results suggest a potential differential inflammatory response associated with CR and exercise, suggesting a potential synergistic complementary mechanism in modulating inflammation. In summary, our findings show that both CR and exercise groups exhibited significantly lower levels of multiple omic biomarkers of biological aging compared to sedentary controls consuming a Western diet. While our results do not definitely prove that CR and exercise slow biological aging, they strongly suggest potential aging mitigating effects. And given the established correlation between these BioAge biomarkers and reduced mortality, and as well as a lower risk of age-related diseases, these data are really, really important. The significant lower levels of several biological age markers among CR participants compared to those on a Western diet align with findings from existing literature, preclinical data indicating that CR not only increase both average and maximal lifespan, but also mitigates age-related disease in both rodents and humans through mechanisms such as improving insulin sensitivity, reduce inflammation and oxidative stress, and the modulation of key nutrient-sensing pathways. Furthermore, the findings that CR participants exhibited lower BioAge compared to exercise participants aligns with previous studies on lifelong rodents and humans, showing that while exercise improves health span and protects against fat accumulation and insulin resistance more effectively than CR, its impact on maximizing maximal lifespan is less pronounced compared to that of CR. Specifically, the lower values of BioAge among CR individuals points to the intervention's impact on metabolic and hormonal pathways that are associated with biological aging processes. Another interesting point in our study is that master athletes exhibited a younger phenotype in DNA methylation BioAge and transcriptome BioAge, both derived from colon mucosa biopsies, closely resembling that of younger sedentary individual consuming Western diet, potentially mediated by exercise-induced anti-inflammatory and senolytic responses. In contrast, CR practitioners exhibited lower BioAge values in gut microbiome and blood-derived measures, highlighting potential tissue-specific differences in biological aging across intervention. A very interesting finding, I think, is the analysis among the KEGS pathways showing uniquely enriched in the CR signature of ether lipid metabolism. Studies in C. elegans link genes in this pathway to longevity, suggesting ether lipids playing a key role in aging. Indeed, in humans, in centenarians, centenaria often exhibit elevated levels of alkyl ether lipids, which are more resistant to lipid peroxidation, offering protection against oxidative stress. So, just to end up, what is the bottom line? Lifestyle changes like calorie restriction with optimal nutrition and endurance exercise can significantly lower biological age, multiple biological age biomarkers, reduce inflammation, and improve overall health in a complementary way. Aging is inevitable, but how we age is something we can influence. So this is the bottom line of this study using the most advanced multi-omics technology and bioinformatic statistical analysis. If you found this video helpful, don't forget to subscribe and share this video with someone who is looking to live their best life. Together we can increase our knowledge on healthy aging, on the secrets of the biological secrets of healthy aging, and this is all for today. See you next time, and until then, stay curious and stay healthy. This is, as always, Longevity Now FL, the channel of the science and philosophy of health and well-being. I'm Luigi Fontana, a physician scientist, professor of medicine, and the scientific director of the Charles Perkins Center RPA Clinic of the University of Sydney, and the clinical academic of the Department of Endocrinology at the Royal Prince Alfred Hospital in Sydney.